at an example. Here we have a drug called warfarin. Warfarin is an anticoagulant, and what warfarin it inhibits clotting. It inhibits clotting. So, but it has a very narrow therapeutic window. You have to keep the concentration of it in a very tight range, or else you can bleed to death. Or, on the other hand, you will have no effect. I had an uncle who had pancreatic cancer, and they put him on this drug for a post-surgical intervention. And it wasn't the surgery that killed him; it was the clot. So obviously, he didn't have his concentration right. And warfarin is degraded by cytochrome P450 enzymes, which are given the designation CYP and then a series of numbers and letters. The primary one that degrades warfarin is CYP 2C9, and there are a couple of other cytochrome P450s as well. And we're also going to look at another variation called CYP 2C19. So we're going to pull a sample from the National Center for Biotechnology Information, or NCBI. And we're going to be using some of the tools, excellent tools that UC Santa Cruz has at their Genomic Institute. They have a really superior, world-class genome browser, which will allow you to access multiple genomes: human genome, rat genome, Neanderthal genome, white rhinoceros genome. You name it, it's there. It also has a really superior set of tools for looking at genomic variants. So here's the sample from NCBI that we're going to look at. It's from a Caucasian female who is a Utah Mormon, and she's a maternal grandmother, so she's probably in the age of somebody who might be a candidate for using warfarin. Workflow is that we're going to look at take the raw reads, put them through an alignment program, do a variant calling, then filter out the significant variants, and then use a visualization program. So here's the loading in of the sample. And you can use any one of a number of different places to load it into the Galaxy program. And you just push the execute button and it'll load it in for you. Similarly, doing the alignment is a very simple matter of picking the right menu items. You pick a recent human genome, you tell the two samples to use, and then you hit execute. And after running the variant program, you end up with something that looks like this, where you have the chromosome that the variant it found is, position on the chromosome, the reference sequence, the alternate sequence that it found, some quality scores and information about how it did it. For example, one of the things that is in that, buried in the info line, is the depth of sequencing. How many times did it find that particular sequence? So the next thing we can do is we can use the UC Santa Cruz genome browser and look at the sequence. after you come into the sequence, the browser, you type in the name of the sequence that you're looking for, the CYP2C9, and it pulls up the gene and shows you all the variants that you found. Here's a listing of all the variants that have been found, and you can see that they're pretty much randomly distributed across the gene. And below it here are what are called the flagged variants. The ones in red are the ones that are most of interest because these are ones where it's going to either change the sequence of the RNA itself or how it's spliced to form the RNA, which is even worse because it makes for marked distortions in the end sequence that's generated. And here is our sequence. And you can see that there are 1, 2, 3, 4 places where there are variants which seem to look like they're in regions which might have some sort of clinical importance. So the next thing to do is to find these lists of relevant variants. And we're going to use a tool called the table browser. So the table browser allows you to go in and say, here are the properties that I want from this particular set of variants lists, and then go in and get an output. The output is basically to another web page, which you can either save as is or do what I did where I dropped it into an Excel worksheet. So after going back to the Galaxy program, hit the other link to the Integrated Genome Viewer, which is a local program which you put on your own computer. And you bring it, and it sends your variants file to the program, and you can see here the whole genome. And now we're going to zoom in. So here we're on chromosome 10 in the region right around the CYP2C9. And you can see that there are a couple other cytochrome P450 genes in the area. There's the 2C18, the 2C19, the 2C9, the 2C8, and the 2C8. So now let's zoom in on 2C8. On the 2C9. And we can zoom in way down right to the level of the single nucleic acid level, which we have here. So here we can see that there basically a little box that says that there's one copy of a normal gene and a variant gene. And it brings up an interrogative which says that this is at position 94, 89, 12, 96, and that the genotype is an A changed into a C, and that the change is a heterozygous one, which means that this person has one normal copy and one abnormal copy, and that the depth of reads that was found is there are 94 incidents of this particular variant found. So looking at this particular location, we find that it is on our list of potentially clinically significant variants. And it's given an RS number, which is the number to look up, of 105, 79, 10. So now we're going to go to another website, which is called PharmGKB. And in PharmGKB, they have lists of all the variants that for the pharmacogenomics that you can possibly want. And we looked up RS 105, 7910, and that is variant 2C9*3. And the *3 variant has a slower than normal metabolic rate, which means that the warfarin would be metabolized slower than one would expect for most people. So this would be a problem. If this Mormon lady were to take warfarin, she would get toxic levels of the drug. So that's to be avoided. So if I were a physician, I would say, well, what's the next alternative? Well, clopidogrel is another alternative anticoagulant. But remember we were talking about 2C19? This lady also has a defect in CYP 2C19. It's the variant *2, which has an alternate splice in it, which truncates the protein and makes it inactive. And that's a problem for her because clopidogrel needs to be bioactivated. So she would not be able to use this drug either because it would not be effective. It would never be activated or activated very poorly. So the only option she has for an anticoagulant is to use a very low dose of warfarin to start and then titrate her up until she gets to a safe level. So to summarize, next generation sequencing uses massively parallel methods to generate an enormous amount of nucleic acid sequence data. Although alignment to a reference genome is computationally demanding, publicly available resources are available to overcome the problem, such as the Galaxy server. And there are many free programs that you can use in Python, R, and other languages that can help you analyze next generation sequencing to generate significant data, which is both clinically and researchally relevant. And with that, I imagine my dear old friend Atlas is getting rather bored, and he's tired of hearing me speak, and I open the floor up to questions.
Hi, Paul. Thanks so much for going ahead and giving us that great lecture. There were a few questions that came in during your talk. One of them was, how do you generate a reference genome? And another is, if you are doing a de novo sequencing of a novel organism with no reference genome.
Well, that's exactly the point I was making at the beginning. If you don't have a reference genome, it really is, it's like assembling a puzzle with the pieces turned upside down. You can do it, it's just harder. It's called de novo alignment. And with that, it's just a matter of finding where the pieces overlap and getting enough of the pieces to overlap that you can generate long continuous sequences. And then once you've generated long continuous sequences, you make those overlaps overlap until you can get as much of the genome as you can. You may never get all of it, but you can get most of it. And in the human genome, there are still some regions that are not terribly well documented. For example, at the ends of chromosomes, there are regions where there are long repeats of telomeres. And those regions are in flux. So they're really not terribly well characterized. Okay, so it is something that it's a major effort, but it is doable.
Thank you so much, Paul. So another question is, could you please explain the difference between coverage and depth?
That is a very subtle problem. Coverage is the number of, let's see if I get this right. coverage is where you look at a region of your sequence, and you look at the number of pieces of DNA, number of reads that are overlapping it. Depth is at a particular point. So you're looking at any given point along the genome, and you want to know how confident the sequence you're looking at is. You say, how many times do I have a read that overlaps it? And that's depth. Okay.
Thanks so much, Paul. I also have another question here. It looks like one of the attendees has a background in computer science, and they're wondering if they should be a doctor to proceed with this course. What kind of background should they have?
Oh, absolutely not. There are people who are, if you are a computer scientist, you're very much in need, because the people who actually program these algorithms are mostly computer scientists, and they're very much into the very heavy-duty math like Bayesian statistics. And there are whole courses on how do you program the sequence analysis, and it is really a computer engineering problem. And if you have a background in computer analysis, and you're interested and you have a knowledge, if you're willing to try, it's something that you definitely will go far with, and you will be very much in demand.
Excellent. Thanks so much, Paul. It looks like we do have a few more questions coming in. I think we might have time for a couple more. One here is, how do you define a sequence, and how can bioinformatics help to find a vaccine for the coronavirus?
How do you define a sequence?
Yes.
Well, a sequence is a stretch of bases, A, T, G, C, repeated over in myriad combinations. And so what the whole point of doing the next generation sequencing is to pile up enough of the reads so that once you've finished and you've aligned them all up, you have good confidence that your consensus sequence is the correct one. And it's also where you check to see where there are variants. For example, in the case of the coronavirus, just today I read that in the San Francisco Bay Area, there was UCSF study where they looked at coronavirus isolates from about 30 people, and they found that there are 8 different isolates from different places. So there have been multiple. The source of the virus was just not a single point source in China. It was from multiple places. Does that answer the question, Butte?
Yeah, I believe so. Another question is, an attendee would like to know if you can suggest some review papers.
There are lots of review papers out. I would suggest what you do is you look up the word next generation sequencing, and you can find a few. And look up the words genomics, or for pharmacogenomics, or sequencing, and I'm sure that you can find quite a few. Another area that's I'm very much, I think is very interesting for genomics is the area called epigenetics, where you can use the ability to find sequences and find where the genome gets modified. For example, one of the things that is known to almost permanently turn off a gene is you put a methyl group on a cytosine. And this basically causes the genome to say, this is a gene that I'm not going to want to ever use again. And the interesting thing about this is that these marks get inherited, and they are reason that there are some cases where stressors in one generation carry over into another generation. You know, Lamarck was not entirely wrong. You know, so looking at epigenetics might also be an area to see the sort of things that you might do with next generation sequencing.
Excellent. Thank you, Dr. Paul. Oh, I'm sorry, I just lost this one. So how much of the alignment process is an inference process?
It's not as much an inference process as getting the most likely sequence alignment. And it's inference only in the fact that you need to go through a logic tree in order to get through this massive pile of pieces. And the way that it's done is by reducing the size of the thing by putting the sequences in little buckets. And then you say, well, of this bucket, I'm going to take some of the pieces out and say that this one is part of this class of genes that's in this location. And the one over here is probably going to be from that one because it has this sort of ratio of nucleotides bases in it. And you know, you start going through and then you find out what the probability of it being any particular one until you come to some sort of consensus.
Thanks, Dr. Paul. It looks like we also have a couple other questions around industry and jobs. I have one here that, as a current bioinformatics student, what sort of projects in regards to performing NGS analysis would you recommend doing in attempts to build up a resume that hiring managers for either academic or industry would be interested in?
From what I've seen of job postings, there are two types of things that there are three things that the hiring managers are looking for. One, they're looking for whether you've handled the next generation sequencing instruments and whether you've actually gone through and taken a sample, shoved it into the machine and gotten a readout output from it. That's one thing that if you can work in a lab and run the machines, that's a useful thing. A second thing that they're looking for is, do you have the skills for analyzing it? For example, can you go through and do an alignment and then generate meaningful sequencing? And the third thing they're looking for is, do you have the ability to do the programming? And the three most commonly used languages I've seen are Python, R, and Unix, or Linux languages. So if you have those languages under your belt, and you can demonstrate that you can use them for something, then you'll be in good stead.
Thanks so much. And then I think the last question that we'll have time for today is, what else is day-to-day look like for these types of jobs for someone who's looking?
What is the day-to-day of the jobs like this?
Yeah.
Well, I imagine that there'll be either, if you're in the development end of things, you'll either be working on programming and documenting how well your programming systems work, or if you're doing experimental work, be doing sequence analysis, you know. And for example, there are people who work in a clinical setting who do the type of work that I showed today. Or if you're in a research lab, you'll be doing the sort of thing that I will show you tomorrow.
Awesome. Thank you so much, Dr. Paul. Janani, do you have any other things to final wrap up?
I just want to say thank you to everyone who attended today's session. It was very exciting, Paul. Thank you so much. And I think he's going to be doing a continuation of this tomorrow. So we hope, I'm sorry that we couldn't answer everyone's questions today, but you know, we will give you an email address that you can, you know, go over and answer. I mean, we will try our best to answer all your questions, and we could ask more tomorrow. So hope you guys will come back tomorrow to hear some more of the sequencing with Paul's lecture.
Great, thank you so much. So as earlier we were mentioning that we did record the session, so we'll go ahead and email that out to all the attendees. And then that also will include the email address for where you can send additional questions that you may have tonight. And we'll make sure that we get those over to Janani and Paul. All right, thank you so much, everyone.
You're welcome. Looking forward to hearing from you tomorrow.
Thank you, guys. Thank you so much. Have a good night.